If you're taking IB, Bio, HL or SL syllabus, then you don't want to miss this. We are tackling an actual exam question regarding B2.2 Organelles and Compartmentalization. So for the first question, it's actually very simple. On the diagram, label a lysosome using the letter L. So you will find the small membrane-bound organelle with hydrolytic enzymes in it because lysosomes contain hydrolytic enzymes and play an important role in the destruction of worn-out organelles and foreign microbes. Second question, state with a reason whether lysosome is an organelle or not. Now this question is actually asking for the definition of an organelle, which are discrete structures that are adapted to perform a limited range of specific functions, and they are also membrane bound. Thirdly, predict with a reason how a cell might be affected if the enzymes present in lysosomes were not stored within a phospholipid bilayer. This question is actually highlighting the importance of compartmentalization, which by the way is something you need to pay attention to in your revision, but is with specific reference to the hydrolytic enzymes in lysosomes. We would say that if it is not compartmentalized, the digestive enzymes in the lysosome will diffuse throughout the cytoplasm, which will result in unintended damage to other cellular components in the cell. Fourth question, describe how ultracentrifugation is used in the study of organelles. Ultracentrifugation is part of cell fractionation, which involves homogenization or blending of the tissue cells before we centrifuge it under high speed to separate out the organelles according to density. So for this question, we would focus on ultracentrifugation and say that it is part of cell fractionation, which separates cell components based on density, and then say that during centrifugation, the densest components will settle at the bottom as pellets, while less dense components will remain suspended in a supernatant at the top. Fifth question, we distinguish between prokaryotes and eukaryotes in terms of compartmentalization. In this case, prokaryotes have no membrane-bound organelles like mitochondria, while eukaryotes contain membrane-bound organelles. It is important for you to give an example of a membrane-bound organelle in your answer. Secondly, prokaryotes have the nucleus and the cytoplasm are not separated, whereas in eukaryotes, the nucleus is separated from the cytoplasm by a double membrane. So 2 points for 2 marks. Lastly, explain how compartmentalization is beneficial during phagocytosis. Now, this is the second question we see about the importance of compartmentalization. So really pay attention to it during your revision. So for this question, we need to know what phagocytosis is. It basically involves a cell engulfing a pathogen, which then forms a phagosome. The phagosome fuses with the lysosome to form a phagolysosome, and then the digestive enzymes in the lysosomes will then destroy the foreign pathogen. So this is how we will answer the question with reference to the structure. So we first define compartmentalization, describe phagocytosis, and then give 2 advantages of compartmentalization for the full 3 marks. IB Bio can be quite confusing and messy at times. Here at BBE, we structure our revision and class lessons accordingly with exam questions side by side so that we can apply our knowledge as we learn them. See you in class!